Good morning and welcome everyone to the Friday seminars at the Museum of Natural Sciences. It is my pleasure to introduce today to Christoph Meyer. Christoph Meyer is the head of the section Statistical Phylogenetics and Phylogenomics at the Museum Koenig, Bonn. We've been co-workers and collaborators for a couple of years. It's been a lot of fun. Christoph, he's very famous for his works. Well, I should start by saying he's a physicist by training, but then he got somehow co-opted into phylogenetics, which is good for us. He's well known for his seminal works on phylogenetics. He's been developing very interesting tools and tests on phylogenomics. For example, the 1K project about 10 years ago was very important for the development of the field of phylogenomics. And since the COVID pandemic, he decided he wanted to work a lot on machine learning, and now, much of his research is applying machine learning to a lot of things, phylogenetics and beyond. And with this, I leave the stage to Christoph and look forward to what you have to tell us.
From image analysis to molecular sequence analysis. I'm head of the section Statistical Phylogenetics and Phylogenomics. So machine learning, how does that work? Machine learning, or traditional algorithms, they basically use if A, then do B, or for each element of a list, do something. That's how you build traditional algorithms. And machine learning, basically, that learns from many, many examples. You give it training data. You give it examples and the solution. And that for many, many items. And that's how machine learning works. So we have example input and expected results. For example, images. We can give it a lot of images, and we give it the classification. These are cats, dogs, apples. We can have object detection. Basically, it learns how to find the coordinates automatically. Or instance segmentation. Basically, for each pixel in the image, it tells us, is this background, foreground, or different parts of the image. We can have any kind of data and estimate some parameters. For example, you can use it to estimate house prices. You can do all kinds of things with that. Or we can transfer language into language. That's what you have with if you use ChatGPT or a translator. You give it language and language. And also the training of these, you give it a sentence and the expected result, and it will learn how to do that. So typically, we need 3 independent data sets. The training data set, which has all the training objects and the training labels. So this could be images and cat, dog, what that is. And we need 2 more data sets, a so-called validation data set that has a lot of validation objects, and a test data set with test objects. And we have the validation labels and the test labels. So this is used for training. And after we trained it, we will use the validation data to see how well it worked. And maybe we try different parameters. We try different model architectures. These neural networks, they can have different layers, different number of layers, different number of neurons in these layers. And that's all what we have with these. These are all called Piper parameters. And with the validation data set, we choose the best parameters. And finally, we have a third data set, which is a test data set, and we use that to test the accuracy. So why do we need 2 of them? Because it could be that our data, or the parameters that we choose, that they're best for this validation data set, but they might not be general. So we have to have another test data set to test the final accuracy, to be statistically independent of what we have seen before. So actually we need three data sets here, the training, validation, and test data set. Very crucial is we have to make sure that there's no leak of the data between the training and the validation and the test data. So they have to be completely independent. You can say, oh, that's trivial, that's easy, but it's quite often that students basically manage to, that it happens that there is a leak here. Just take this example here. We have images in a video, and you take frame 1 for the training and frame 2 for the test. Probably not much has changed between frame 1 and frame 2, but you quickly have a leak from the training to the test or vice versa by this. So I've seen many cases before, and you have to be really careful about this. So avoid leaks. This is one of the few things in life you have to be really paranoid about, that there is leak between these data sets. Okay. At the same time, the training data set has to include the full variety of characters of the objects that shall be classified. That's also important. If you design training data, it has to have the full variety. And that means, for example, if we classify flowers, the flowers have to be positioned at all parts of the image equally. So they have to be uniformly distributed over the image space. We have to have all sizes of flowers in the training data. We have to have all color variants. We have to have all foreground-background combinations. And we have to have all orientations. For example, if all flowers look to one side and we want to predict them and they look to the other side, that might not work. So we have to have all orientations. And then we have to have all combinations of the above. That's also important. So and that's how our training data has to look like. If we have a flower, for example, if white flowers never occur on the left side of the image, we will not be able to detect them on the left side. The neural network is not that smart that it only learns that flowers can be everywhere by showing these flowers everywhere on the image and in the training data. That's very important. Every variation or combination of features a neural network has not seen, it might not be able to classify that or see that. That's very important when constructing training data sets. Of course, it also tells us they're not really smart at that point, but we have to show them what the variation is that they should be able to detect. How do we deal with that? Because that is already a problem. We might not have that many flowers, images of flowers, where the flowers is in the left, right, lower, and in all the corners, for example. So we have to create new data records. This is basically called data augmentation. We create new data records that have been artificially generated from the original data, for example, by flipping images left and right, by resizing the images, by cropping the image, and by masking part of the image randomly, by changing the brightness and the color saturation and partially mask images. And actually, one very advanced thing here is to use neural networks to create new images with a combination of features. These are generative neural networks, like the ones you've seen, artificially generated images, art images. And they can also be used for this, what is called data augmentation. This is very important. The technique that we typically use today is what is called transfer learning. Transfer learning, these are neural networks that have already been pre-trained. That's a very interesting concept. So we do not train image classification or object detection, landmark detection neural networks from scratch, but we fine-tune existing neural networks. And How that works is as follows. We have a pre-trained data set. This is a neural network that has already been trained, maybe on millions of images. That's a typical thing. If you have these plant identification apps, they have been trained on millions of images. And what they consist of something that encodes the image in so-called feature maps. And then we have what is called a classifier neural network here, and they classify this into different objects, cats, dogs, and whatsoever. And this part here, that has been trained on millions of images. And now if we want to apply this or train this for our own data, what we typically use is that we cut out this part with all the trained neurons. And we add a new classification part here. So these are new classification layers, and we only train these. So all the features that can be detected, edges, round circles, all these image types, they are encoded here in this part, and this here is what is trained. That's very interesting. So if you would have someone who is an expert on some species, we could maybe make a copy of his brain, of that part, and use it somewhere else. And that's what we are doing here. So we take that part of the brain, basically, copy it here, and do the classification separately. This is what is called transfer learning. So we actually just add a few more. We remove these layers here, add new layers for the classification. That could even be a different number of classes here, and then we can train that. That will be more accurate and faster in training this. Okay, there are quite a number of pre-trained models out there that you can use. This is just a very quick here from MathWorks 2022. They compared some of the nets. Here, this was the first AlexNet, that 2012. That was maybe the first well-known deep learning neural network. This here is the number, the size of the circle represents the number of parameters. This is the accuracy on some of the data sets that are typically used. You see, these are more complex models, and this is the relative speed that is required on a GPU. That's not an absolute number, this is only relative here. So in biology, we are working with image recognition, object detection, and image segmentation. More recently, it became also basically, we also want to, or it became popular to do landmark detection for morphometrics. There are also, we also apply this for multiple sequence alignment, phylogenetics, sequence classification, genome annotation, and protein folding. These are also applications of neural networks, typically in biology. Some selected projects here. This is a cooperation with Marianne Espeland and Sneha Bansali. She's now in Frankfurt. We classified butterflies here. That works pretty well. We can also use it here for image segmentation. Image segmentation is cutting out the background here. So this is the segmentation mask and the segmentation image, but we can also use it to extract different features of the butterfly. We can train it to only find the wings and extract everything else. We can train it to only find the back wings or the hind wings or the forewings. So we can train a neural network that extracts these features. So these are the forewings and back wings. So we can even train it to identify the spots on the wings here. So these are, zoom in. This is the original and we zoomed in here to see these parts more clearly. And this was trained to identify these here, these spots here, and only on the forewings, not on the hindwings. And the neural network does that automatically. So you give it the original image, and it will give you all these forewings for each of these. And that's really interesting. Here you can also have this for these spots. And now you can do measurements on these spots. So this is really interesting for morphometrics, for example. So the use of shape for morphometrical analysis. Especially for image segmentation, we need a lot of images. So here we need typically 100 images per class or more, maybe even a few hundred. They have to reflect the full variety of the backgrounds and all morphotypes in all combinations. And they should be without systematic biases. That sounds easy, but it could be difficult because we have to generate a lot of training data. This is another project here. We identify beetles. We classify edigus. Edigus, these are the reproductive organs of male arthropods. And they look very different. These are 3 different species here. These are habitus images, and these are the edigus images. It does not always react here immediately. Okay, so here we have 3 different species. This here is the scale, so that's 0.5 millimeter here. So that's pretty small. These are really small beetles. Okay. In this case, image segmentation is quite important because if you have different backgrounds, that can basically mislead in the classification. If you have, especially if that is systematically dissimilar. Maybe you take one set of images today and another one tomorrow, and they're all a bit brighter today than tomorrow. Then you have a systematic bias in your data set, and you should remove that. First thing is, if you remove the background, you're already in a much better position, and then you should make all the foregrounds equally bright. That's not very difficult. You can normalize that, but that's something you should take care of. So what we used was image segmentation with the segment anything model. That's the SAM model. Facebook has created that one. Basically, that can be used to segment images out of the box. You just give it an image and it tries to find the different parts of that image. We can also use self-trained models. Basically what we do, we fine-tune a pre-trained segmentation model, for example, from TensorFlow Hub, and we can work with that. This here is what segment anything does. You just give it the image and it tries to find the different parts of that image, and it does that surprisingly well here. So actually, for our beetles, it typically produces many, many parts of the image. And typically, one of them is the beetle that we want to have, or the reproductive organ. Sometimes it's a combination of them, but typically it's not very difficult to basically combine them. You get typically quite good results. These are some segmentation results here of the reproductive organs here. You see that works quite well here. They are all glued on a piece of cardboard here, so that's what we see here. And the neural network has been trained to identify these here. Actually, this has been done with segment anything. We also use them to train, to generate training data for self-trained models, and basically to improve results. So if that doesn't work perfectly, the automatic segmentation, we can improve the segmentation mask and train our own network to get even better results. So here are 3 types of images that we looked at. We looked at the raw images. That's not the same image here. This one here is a mask image, so we only have the foreground and basically the silhouette. We can look at the reverse masks. This here, we only have the background and the silhouette. And we have only the masks. That's only the silhouette here. We also have to do data augmentation here. Actually, data augmentation and classification has been done with the YOLO. I don't know if you have used machine learning before, you might have come across YOLO. YOLO is a framework that YOLO stands for you look only once, because and that was the original aim, that you don't need a lot of training data. You still need a lot of training data, but that was the original name here, or aim here. And what we look at here is the augmented data set that is produced by YOLO. I told you before, we want to create artificial images to enhance our data set. And these are the images for our beetles here. So you see this one here, they look all very different. This one here has been, you see, you have to be zoomed in, and part of the image is removed here. This one has been moved to the back. It used to be in the center here, or it has been cropped, basically. This is how augmented images look like. If you have questions, maybe, should we answer questions immediately? At the end, okay. We also see masks here. So in order to make the model more general, some parts of the image are randomly masked. And we try to train it. It might fail on these here, because in some of them, really important parts are removed. But altogether, it improves the classification results of this neural network by adding these here. And this is actually a really, really important part to make the model more general. For our beetles, this worked quite well. We have 114 classes, which are 114 species. The Adigus, for the Adigus, we have 1,397 images, and for the Habitus, we have 925 images. You can immediately see this is only less than 1/10, less than 10 images per class. This is very little. I told you before, and if you look in the documentation of YOLO, it says a few hundred would be good. But often, many less also work. So with these numbers here, we get an accuracy of 95.7 basically for the Adigus. On the raw images and on the mask images, we get 99.2% classification results. For the Habitus images, they are more difficult, but we still get 96% or almost 97% here. We also have fewer images here, so that works quite well, even though we have basically, I would say, too little training data. This is another project with Koma Kotra. She was a master student, and we detected insects on flowers for monitoring. Here you see the crown truth, and that's what the neural network predicted. That's basically the test data set here. You see that this coincides. This is not identical. Actually, I deliberately chose an example with a few errors here. Normally, it works quite well, but it's not 100% perfect. Here you see that this fly should be a bug, and this bug should, this is the crown truth. This bug has been identified as a fly. These have been identified properly here. So the prediction success is quite high, and we can track insects over time. So we can do, oops, it should, does it play the video? No, it doesn't. No, it does. You see there's a wasp here. If you would basically zoom in to that video, you would see that this is a wasp. you can track, you can monitor insects in that way. Okay, that's this one here. Yeah. Sorry. We also use this for automatic landmark detection. Landmarks are quite laborious to label. So here we have fish images. This is actually a cooperation with Madeleine Stange and master student Erika Sirelles Dupuis. She already left the museum, or she finished her master thesis. So this is one of the fish that has been landmarked automatically. It's not 100% perfect, so this one should be, I think, a bit further below here. Some of them are just slightly moved here. Altogether, this worked reasonably well, but it was still lacking the perfection that we thought we would get. You see it worked well, but on some of them, you might put them, yeah, this one should actually be placed here. It's quite well. A disadvantage is that you still need a lot of training data for these here. This is a really difficult task for machine learning, it turned out to be more difficult. Yeah, this is also what we use neural networks for. Machine learning can revolutionize morphometrics and taxonomy by using all these methods here. Let's come to sequence alignment. This here is what we already saw. This is image segmentation. So we can train a neural network to look at images and to say, this is the cat, this is the grass, these are the trees, these are the sky. This is what these image segmentation neural networks can do. We had this with the butterflies and the four wings and the dots. And the idea was to use that also for, basically this could be seen as an image here. That's a sequence, 4 sequences here, and 3 sequences. And the alignment could be seen as the image that we want to transform this into. And so we use these segmentation neural networks and just applied them to sequences. And that works quite well here. So we give it this here, these sequences, and it produces that alignment here. So that's something that we do in my group here. That was a project with Peter Arsik, and we published that as a preprint here. And so we have neural networks that can do this. Actually, they perform quite well. They do have some limitations. At the moment, they only work for fixed size matrices. So at the moment, they only work here for 48 by 48, and that means 48 nucleotides, 48 sequences, or 96 nucleotides, 96 sequences. They have been trained with specific gap distributions. And we found out that if we have different gap distributions that deviate from the ones that we use in the training data set, they start to hallucinate, actually. They start to invent things. That's what you have in ChatGPT. We try to make these neural networks more human-like, and now they make the same mistakes that we do. So yeah, but it works quite well here for the cases, for the gap distributions that we built this for. It's better than, that's our AliUNet, that's Clustable Omega, that's the MAFT software, Muscle and T-Coffee. You probably know these alignment programs. And this is the accuracy. And so this is more accurate than the other programs here on these gap distributions that they have been trained for. So in some cases, it hallucinates bases. In general, normally it does that to improve the the sequences so that they're more similar. So it does that in the right direction. But this is a novel approach, and with more training data, we're quite confident to get much better results. The amount of hallucinated data is in the order of maybe 2%, which is still high, but we try to improve that. We are also using it for that's not sequence alignment, maximum likelihood method. This is a project with Nikita and Iker. So we already published the methods here a while ago. This is a tree reconstruction. Maybe I should have put some more images here. We have 4 sequences, and we want to identify the best phylogenetic tree for 4 sequences. And that phylogenetic tree can be estimated. Normally we do that, for example, with maximum likelihood. Maximum likelihood looks at all the sites and finds the most likely explanation for that tree. We had the idea to use machine learning for that. Basically, take the site patterns. Site patterns are, if you have an alignment, the first position in the alignment, second position, these patterns that you have. And you can classify them to, the frequencies of these can be classified to get the most probable topology. And that's what we do here. And we use different models here, the Chooks-Cantor model, the Kimura model, Felsenstein model, HKY model, and the GTR model, the most general time reversible model. And these are the accuracies. This is neighbor joining, that's maximum likelihood, and that's the neural network. So the neural network is quite close or equally good to what we can do with maximum likelihood. The big advantage is that the computing times are much shorter than likelihood. Likelihood is basically there are proofs, mathematical proofs, that we can't be better than maximum likelihood. Then of course, we can ask the question, why do we look for something if we can't do better? But we could be much faster. Maximum likelihood is very computationally intensive. So, and if we can improve that by a factor of 1,000 maybe, to just take 1,000th of the time to reconstruct a topology, then this is a big advantage here. It's about as accurate, but it's much faster than the maximum likelihood method. Actually, at the moment, we are working on a joint project here. Nikita and Iker, Iker is the first supervisor on this project, and we are working on this to improve these methods even further. And that's a really, I'm really happy to be in that cooperation with you. Okay, we can also use this in ecology machine learning as a general thing. Now I want to give some outlook here, what it also can be used for. So what we have here, you see that we applied here what is called object detection. We have these images, and we automatically identified butterflies and flowers here. Butterfly and flower, a butterfly. There are no flowers here. Here is a butterfly and flowers. Butterfly. Okay, and what ecologists are interested in is, for example, species interactions. Here, pollinator, plant interactions. Which insects visit which plants? And that's something we can do automatically with machine learning. We give it a lot of images, and it will identify the species, the insect, and the plant automatically. So we will see here, these are certain flowers here, and there's an interaction between that butterfly and these plants. So we can build what is called interaction networks here, and machine learning can be used to automatically identify a large number or interactions on a large number of images here. Another possibility is to use large language models, for example, ChatGPT, to extract this information from the literature. For example, if you look at this, this is a page from Darwin's main work here, and you see here he says, of these here, the two species may have often been seen on other species of climbing about the flowers. So these here, they visit these flowers here. They visit these cactuses. You can extract these species interactions from the literature. They have been described many, many times. And you just have to go through scanned books and see whether you can find these species interaction to build what is called a species interaction network. And that's what ecologists are interested in. Ecologists, they have these interaction networks where they have one species and the other species and all these connections here. And that's very important, for example, if you're about to lose one of the species, you also lose all the interconnections, and maybe you don't have pollinators anymore for another plant, which might have other consequences, because that plant might be visited by other insects, for example. Their larvae might put their eggs onto that plant. And that's very important to document what are the interactions in nature. And these interaction networks can be built basically with machine learning. What's the time? Do we have? It's almost over. Okay. This here is another example where we can use machine learning. There are these biotic indices in ecology. You count certain number of species and you say, this is a very rich habitat, this is a very poor habitat, almost everything is dead here. So if you have these here, the stone flies, dragon flies, these are really good. If you only have these worms, that's very close to no life. And that's very important for ecologists to determine the quality of a habitat. And what has been, so state of the art is basically you take a piece of soil and you count the species in the soil. But that's also very time consuming. People want to have it more quick. And an alternative is to use eDNA metabarcoding. eDNA typically counts a number of bacteria or very fundamental organisms, small organisms. And of course, these biotic indices, they're not the same as what we get with eDNA, but we can connect that with machine learning. Basically, we can, that has been done actually here in these papers. Cordier, Tristan Cordier, 2017, 2018, that he has used that. They counted specimen, determined their biotic indices, and they also used eDNA and did a model training to predict the biotic indices using the eDNA data. So there must be some kind of, or it's likely that there's a correspondence between the number of specimens that you have and the eDNA composition. And this transition here can be used, basically you take the eDNA, you take the biotic indices as the label, and you do machine learning to do this transformation from eDNA to biotic indices. And that works quite well, as been demonstrated. So no biases were found. This should actually be possible for a large number of applications like toxins, other factors, that impede the quality of a habitat. For example, that might, oops, I pressed the wrong button, sorry. That might be the perfect habitat here, a healthy habitat. And if you have substance A maybe, or stressor B, the eDNA composition changes. And you can do machine learning to transfer this to basically give a diagnosis of what you have. And that would be a very that's a very interesting application of machine learning as well. Actually, these neural networks would be very close to the ones that we are using here. You classify this distribution into certain habitat types, certain toxins, certain problems in the data set. So eDNA, a machine learning classifier, and you can predict the stressor. So the advantages are it's much easier, much faster. You use eDNA methods. It's comparatively flexible and scales well with a large number of data sets. These are the big advantages of these here. So this here has a hidden variables in a dynamic Bayesian network. So these ideas here, they are not used very often at the moment, but they have already been published a few years ago. A problem with machine learning generally is the quality control. Typically, it's not really inherently inside the method. You have to do some extra work to do the quality control of machine learning methods. That's a very general remark here. But a lot of people are working on this, and I'm quite sure that we will have more. There are, for example, the generative adversarial neural networks that can be used to basically incorporate the quality control into the workflows. Okay, I think. So these are the central takeaway messages. We can use it for morphology, sequence analysis, rating, classifying habitats, and ecological system analysis. That's where we can use machine learning. That basically covers most of biology. Or I have been able to talk about, it could be used even broader, but this is a big summary here. So many thanks to my colleagues, Nikita, my other students, Iker, Marianne Espeland, Madeleine Stange, Henrik Krenwinkel, and my former PhD student, Marie Brasseur. So I think we can come to the, many thanks for your attention, and now we can come to the questions.
Thanks, Christoph. It was a really interesting talk. We have now time for some questions. I think Christoph gave many ideas of applications of machine learning for many of the things that are happening in the museum, not only for people doing genomics. So that's very interesting.
Hi, thanks a lot for the talk. I was wondering, in my group we work with behavior with birds, and we record the nest while the parents feed the chicks. I was wondering if it would be possible to train a machine learning algorithm to extract that data, like the parent is here, is feeding this individual chick, this chick here is begging with this intensity. Would it be possible?
I don't understand.
Use machine learning for, igual puedes gritar un poco más.
Feeding behavior in the nest.
In birds.
In birds.
Between chicks and older birds. you can identify the interaction between the parents and the chicks.
That should be possible. It's also used in behavior a lot. People use it, for example, to track mice in environments and see how they interact. This is already used, yes.
Yeah, but as far as I know, for this kind of videos, you need to have a consistent background, like always the same type of background. That's not the case for my data. It's always different. I don't know if that would be a problem. I would require more data.
I haven't seen it for birds, actually. That would be something new, maybe. I think it would be interesting to do that. Does that answer the question? Yeah.
Thank you.
More questions?
Hi, thank you, Christoph. It was a lecture for me instead of a talk. My question is directed to the using of machine learning for taxonomy. So what if you have an image or a group of images of a species that is not in your training data set? So there is a new species, for instance. How the machine learning or the method would deal with that kind of images if they are not in your training data set, if it's a new species or different, how it will classify it?
When you do image analysis with for taxonomy, what happens when you have a species that is not in your data set?
Will it incorporate it to the training data set and learn from it, or?
Yeah, of course that is a prerequisite. You need the specimen in the data set. There are machine learning methods that can identify outliers, but that is also something that is non-trivial to identify outliers, because how do you characterize an outlier? We know what an outlier is, a completely different species, maybe, but it's difficult, yes.
Because I'm struggling sometimes. I'm working in a project now also of this, to use a machine learning for classifying specimens with a taxonomy.
It's comparable to barcoding. If you have a barcode of something that is not in your database, you get the closest match, but it might, you will see that it's not perfect.
So does it identify it as a different class or outlier, as you said, right?
Yes.
Yeah.
Okay. So I sometimes move a bit back. I don't know whether the camera still catches me.
Okay. Thank you so much.
Yeah, basically that's that's a problem. Yes, if you don't have that in your data set or in your database, and it should be in your database to work with that.
Thank you.
More questions?
Thank you, Christoph. I'm going to first follow up on her questions. So we've been using a machine learning method to classify arboreal and tropical species. What we did is to use like a hierarchical classification. So if the species was not there or the model was not able to identify the species, then it could identify to genus or family level. So that's something that some machine learning methods can do. My other question is unrelated to that. I was wondering if which large language models are out there that could help us extract that key text that we want to use for whatever. Like it can be interactions, but it can be also trait values or whatever. Because you just mentioned large language models, but I'm not sure if there's something out there that is already quite good at it. Or...
You can try to use large language models to extract all kinds of information. For example, you can identify text passages where species are described. You can find text passages where it's described where they live.
Yeah, but which ones was names of large language models, specific ones that you've maybe
that can
used?
Of course, you might need a lot of training for common names, scientific names, maybe different versions of that name in history. But in principle, that should be possible, yes.
Okay, but do we have to develop? I mean, like if I want to use it for a specific task, like extracting, and let's talk about interactions, extracting which bird is eating what kind of plant or whatever, has anybody already developed this kind of large language model, or we will have to train it ourselves?
To some extent, you can use existing large language models. For example, if you have a piece of text, which might be a few pages, you can put that into your prompt. You know what, if you use large language model, what you type is the prompt. And what many models do is that they add some additional text. You can even influence how they work. You can say, be very friendly, and these large language models, if you put that in front of every text, they try to be extra friendly. You can also give it two pages of text and ask, can you extract this and this information from that text? You can do that, basically combining text and your own question into one prompt. That can give you good results without training them. Of course, if they don't know they have to know the vocabulary, basically. If that vocabulary has not been trained in the machine learning model, in the large language model, it will not be able to recognize it, but it has been trained, and the big models like ChatGPT, and they typically have been trained on basically everything that exists.
Yeah, my question is whether ChatGPT will be like good enough to actually do the task all right, or would it be missing information?
There are also, for example, BAT. BAT is a model. There is also BioBAT that has been trained on a lot of vocabulary specific to biology. Actually, it basically has been fine-tuned for this task, and that would also be a possibility. There are existing publications that the ones that I showed here, these are quite new ones. They're not the original ones, but they have all the references inside that can be used to basically see what has been done in this field.
Okay, we have time for one last question, quick one. Otherwise, we close here. Thank you, Christoph, again. Thanks, everyone, for coming.